Hi, good morning, and welcome to the ZP Developer Zone live streaming webinar. So, in fact, we are doing this live streaming today. It's been a little while, but we've actually updated the internet connection. With that said, today I'm going to talk about PEDOT electrodes in microfluidic channels, probably how to functionalize screen printed electrodes to make the PEDOT, which is not super rocket science, but also then how to maybe put them into a microfluidic channel. So that's one of the questions that has come in during this week. One of them is just making some comments on doing RNA detection using oligo nucleotides. So I will talk about that today. Also, there's a nice inquiry that we had from India. I think it was from India, but it was via ResearchGate, where they were asking about they're getting a lack of cyclic voltammetry activity on an electrode after they've put down a polyluminol conductive coating, and I will talk about that today. It's probably worth saying I'm also going to do a demonstration as well. So in front of me I have the SenseIt platform. The SenseIt platform is both a device for people who want to make products, but also a potential stat in its own right. It is very QR code driven. So what that means is you can scan a QR code to do something like cyclic voltammetry or square wave voltammetry or linear sweep voltammetry, or you can scan a code also to do something like chili detection or potassium detection, etc. So it's a device that can both be used as a research tool, but also in either taking technology, or when I say technology research, beyond technology readiness level number 3 and taking it higher. And I think this is a way to publication as well, because I always thought it was a way to commercialization, but I am getting some feedback from academic colleagues that actually a lot of journals these days are actually looking for a bit more of an impact, and therefore being able to come onto a technology that's much more like a product and actually demonstrate that you can sort of run it through an app and get real results back on the phone may be useful as well. With that said, let me go back to the slides. So PEDOT on electrodes in microfluidic channels, detection of RNA using oligo nucleotides. Why is somebody not getting cyclic voltammetry for ferri, ferrocyanide through a polyluminol conductive coating? Also, somebody's interested in measuring the oxidative potential of organic molecules, and I am going to do a live demonstration of that today. I've actually got some in fact I should have put the structure down, but I got a very drug-like molecule in here, and I'll do that experiment here. And then it should be worth saying that this is an aqueous solution. A lot of people who think that they're going to need to deal with organic molecules think that they would need an organic solvent, and I haven't linked to it, but at ZP we do have a range of screen printed electrodes that are organic solvent resistant. But if you're looking for a low cost option for measuring electrochemical properties of organic molecules, and you can use these electrodes, and I wish I'd done a better job of actually linking to them. And with that said, you can do it in aqueous systems, let's say. All right, connectors for ZP's screen printed electrodes, I will discuss that today. Also just some nice data that's come out on ZP screen printed electrodes versus glassy carbon type electrodes. And also, I think I said here finally pH sensor, or not, this is penultimate. pH sensors for concrete. Concrete, I think, can be quite alkali. And its pH probably changes during the curing process. So, talk about that continuous monitoring. And then gold PCB microneedles. So there's a lot to go through. I won't really stop. Let's go forward. Just to say that if you're interested in commercialization, or even just that question of bringing your technology readiness level from 3 to higher, please come to this webinar on the 24th of September. There's always slides, and there'll always be a live demonstration at the same time, the live demonstration on the SenseIt platform. If you're interested, I haven't got any wearables on me, but if you're interested is actually more in a wearables technology, then we do have our live streaming webinar on the 1st of October. We always do 2 webinars on any particular on a day, just because then we can cover both time zones. We do one in the morning, one in the afternoon my time, because then that means that we can cover Asia really well with the morning one, and we can cover the Americas really well with the afternoon one, for example. If you're in Europe, you can kind of benefit from being able, both of them will work quite well for you. If you're in academia or interested in how ZP technologies can really impact the translation or the impact factor of your research, then please come to this webinar on the 25th of September. Again, there'll be two parts to it really. There will be talk and notes and not lectures, but there'll also be a live demonstration as well. A lot of what I'm going to talk about today is about potentiostats and screen printed electrodes. So at ZP we are super biased because we do produce our own screen printed electrodes. Just about all of these will be covered today. Just out of interest, if you've ever got an opportunity in life to decide between a gold electrode and a carbon electrode, I would generally suggest always start with a carbon electrode, and I'll talk about that today. And then on the potentiostat side, I do have this SensiOr potentiostat in front of me. This is a really different type of potentiostat that, as I say, it doesn't leave people stuck at the kind of research phase. Actually, it will actually take them through to something that they could otherwise deliver to, deliver to end users. It's much more, the capabilities that's built into it is much more like a product that you would give to when I say real people who are not necessarily scientists in the lab or not. So it's a very user-friendly experience. With that said, first question that came through was, or one of the questions that came through was, somebody wants to put PEDOT on an electrode in a microfluidic channel, and they're asking, in some ways how to do it, and then secondly, companies that can do it. Well, companies that can do it are ZP. The two electrodes that I would suggest for this would be our hyper value carbon electrode, and I think it's called our 501 carbon electrode. I mean, there's, putting PEDOT on electrodes, has been done for, I think, at least 2 decades. I mean, this is something that was going on when I was, doing my PhD. So this is this is the monomer for PEDOT, and this is the PEDOT itself. So I won't let's say, play that, but we have the monomer. If you're using ZP technology like our SensiOr platform, you can actually do cyclic voltammetry on that, and that cyclic voltammetry then, I'm sort of suggesting that you take a drop of your monomer, put it on our electrodes, either one of these will work, and do a electropolymerization. Of course, in my biasness, I would like you to use the SensiOr platform. The nice thing about using the SensiOr platform is that it's much easier for me to be able to support people because I can see what they're doing, or I understand the instrument, and it's both its strengths and its limitations. But of course, when you're using other people's potentiostats, it's hard to then comment on your science because obviously what that potentiostat might be doing under the hood might be actually quite unknown to me. With that said, you can do the functionalization of that PEDOT just using the SensiOr platform. It's QR code driven. If you're interested in that, I've got there's 2 webinars, one on the 24th and one on the 25th. Either one of those will cover this QR codeness, let's say. And cyclic voltammetry is one of those methodologies, and you've got all these settings. Once you've got the hardware connected, you can then change all the settings and have that control of that cyclic voltammetry. They wanted to put this PEDOT electrode actually into a microfluidic channel. So I've got a couple of slides here. I think they may have already seen these slides, but we do put this exact screen printed electrode that you see in the image into microfluidic channels, and we can do it quite a bit. You did ask specifically, what companies can do this, and so ZP is one of those companies, but we would do this as a project. Eventually, we'd like to be able to turn this kind of stuff into a product. But the thing with microfluidics is everyone's always got a slightly different requirement. So making a standard product out of it is actually quite hard. If people are interested in sending me a sketch, I can sometimes help them because the sketch will if I can see the sketch is quite generic, then that might help me be able to sort of, rather than turn it as a project, I can maybe then use it, then maybe offer it as a product. But that said, putting PEDOT on a screen printed electrode, at least 20 years it's been done. You can use the SensiOr potentiostat to do that. You just need the monomer. you'll see me do a demonstration shortly using the Sense It All platform. You just need to put 50 microliters probably onto that electrode and do some cycles, and that's electropolymerization. Then you can mount it in a microfluidic channel like this, and you're sort of done. But actually this last part is the tricky part, because without really understanding your channels, and I mean a sketch with some numbers on it, doesn't have to necessarily be an engineering drawing, but a sketch does help. I think I'm going on to, so take one of our electrodes, put your monomer solution on there, electropolymerize it. I know you're not going to use necessarily our Sense It All platform, but I would like it if you did. But then to put those electrodes into a microfluidic channel, at the moment, with the information I've got, we would have to treat it like a mini project. So at ZP, we do micro projects, mini projects, then proof of principle, and then we do very large projects. So it's just a small project for us. What to purchase. So here's the electrodes that I suggest, and here's the potentiostat that I suggest. I realize in this webinar today that I'm actually not putting enough links in, because there's things I'll talk about that are not necessarily linking to, so I apologize if I'm doing that a little bit. With this one, I'm just having an online conversation with somebody who's going to be doing, I think, some RNA detection in the end. They're probably going to end up using our carbon electrodes and probably some of our gold electrodes. If you've got a choice in life and it's the beginning of your, let's say, research, I would definitely suggest carbon. I think gold is, I can't remember if it was 24,000 times or 28,000 times more expensive than graphite. It's just simple. So when people often, especially in medical diagnostics, say, I'm going to make a low cost, easy to use medical diagnostic. Well, the first comment there is gold is not low cost. So if you want to sort of start with that, if you have that statement in mind, then I would actually suggest carbon. I know why people use gold a lot, and I'll have some slides in a minute. It's the self-assembled monolayers that that's probably 30 to 40-year-old science at this point. It's very robust. I can see what, so there's a big body of literature. I actually suggest that carbon's probably better if you're eventually wanting to actually translate this out of the lab. The way we would do it is we do exactly this kind of molecular assay quite a bit at ZP. We've had a program running recently where we've at least done 1,000 hours worth of work on a similar thing to what I'm about to discuss. Take the electrode, put some micro drops along there. The micro drops will coalesce. So that's kind of how you can get the kind of SAM layer down. Before you do that, clean the gold surface with about 10 millimolar HCl. Don't spread the HCl over everywhere. With solutions, always put the solution where you need it to be. So if you want to activate that gold electrode, put about a 10 millimolar HCl, hydrochloric acid solution on that electrode. Clean it and then rinse it with DI water. That will make the surface pristine. And one of the things that's not talked about much with gold is it really tarnishes quite quickly. So even if we make a pristine gold electrode, it's always picking up stuff from the atmosphere. So by the time you're using it, it might not be quite so pristine. So do some research into activating gold electrodes just before you do the SAM layer. So we activate our gold electrodes just before we do the SAM layer. We don't just put the thiolated oligomer down. We actually space them out with MCH, with I think it's mercapto. I want to say mercaptohexanoic hexanol. I think it's mercaptohexanol. So we have long, let's say, capture probes and short spacing probes. They're both thiolated. Otherwise, you can't really pack all those oligomers next to each other. You do have to have a certain spacing. The reason I'm being a bit fluffy on what the ratio between spacing and capture probe actually is it really sometimes needs to be optimized. We've recently sort of been reading literature where actually you can try certain things, like sometimes we're trying a solution at the moment of capture probes where the concentration of the capture probe is, I think, about 1 micromolar, I want to say, and then 0.1 micromolar and 0.01 micromolar. As you can see that we're trying quite different concentrations, but that still hasn't answered the question, what's the ratio between that spacing, the short spacers, this MCH and the oligomer and the capture probe. And I think you really have to kind of optimize that part. And so my recipes might not work with your target. With that said, we often do a co-deposition of these two, and as long as the gold is activated. The other thing that people sometimes don't realize is that when they get these oligomers in, they're actually in the disulfide state. So make sure that you've done a reduction of the disulfide to the thiol or the sulfide before you do this. I have seen academics who haven't realized this and actually tried to activate the gold surface with a disulfide version. So you do have to reduce to make sure everything's in the thiol state, and that's a chemical reduction rather than an electrochemical reduction. With that said, then you can form these SAM layers. They're quite robust. Reading what these people are wanting to do, they're then going to essentially capture the target, which is kind of straightforward enough. Or at least the way I show it, it's quite straightforward enough. And then they want to actually essentially show that the target is in place. So I think the way they're planning to do it is to have these signal probes which have methylene blue on them, and then when it binds, they want to show a kind of change in signal. I have seen this kind of assay work a little bit. There's some company called GenMark DX in Carlsbad, California, and they do something similar to this. In fact, at ZP, if we were to do this, we wouldn't so much do this. We have done it this way. We are actively doing something very similar to this at the moment. But I've got some other slides in a minute, which are more immunoassays. But strategies I think you can use in immunoassays are also strategies that you can use in molecular assays as well. So watch my few slides in a minute on immunoassays. You can do this signal you can get this signal out of this using linear sweep voltammetry, square wave voltammetry. I think these would be the techniques that I would be trying. Oh, and also EIS as well, electrochemical impedance spectroscopy as well. The reason I show QR codes is because I'm also making the point that most of what I now talk about through these webinars, especially can be used on this platform, and I can see that on this platform we do have the QR codes for square wave voltammetry, cyclic voltammetry, linear sweep voltammetry, EIS. So these kind of techniques would work well in the kind of assay that these people are looking to develop. With that said, let me go forward slightly. Yeah, so EIS is in here. So here's some EIS that I did actually. It's not a molecular assay, it's an immunoassay, but an immunoassay is basically a binding assay. Here we're using EIS, so as we get increasing binding, in our case, the signal was sort of changing in this manner. So if I choose one of the frequencies, we can plot frequency versus concentration. I've essentially come up with a calibration curve that way. You may also want to use square wave voltammetry. Now, admittedly, this is actually cyclic voltammetry, but I just want to show you, that you can get a signal versus concentration, plot that out, and you've got yourself a kind of essentially a calibration curve. The reason I bring that up a little bit is because I'm obviously encouraging people, and again, a bias has to be put out there that, we do manufacture this at ZP. Again, it does help if people are using this, and especially if they're then asking questions, because I'll probably know the answer, whereas it's coming off other potentiostats, which I've probably owned and used over the years, but I do think that these other potentiostats now are holding us back in terms of being able to translate science. This particular potentiostat does come with a data pipe built in, so you can put the sensor on there that's been functionalized. Eventually you can bring the analyte to the solution. You can run the particular method that you want to do, and what you'll find out is it will record the raw data and send the raw data to a cloud system called Julie. If you're unclear on this, come to the live webinars. As I say, the next one in the series is on the 24th, and there's another one that's quite similar on the 25th of September, so not that far away. We can process the data in that cloud, and we can actually give you a information back, like a concentration, but only if you've got the calibration curve. So in the end, the let's say the problem that we're trying to solve is, and in fact I'll describe that problem in a minute. I just want to give you another idea that you can use carbon electrodes to also do immunoassays. I'm using you can use carbon electrodes to do immunoassays. You can also use carbon electrodes to also do molecular assays as well. So this is an old paper from me. I have talked about it a lot, but it does actually describe a simpler way, let's say, of doing an assay. We take the electrodes. In this case, we're immobilizing using pyrene NHS. There's an acronym for pyrene NHS, which I have recently seen in posters. I can't remember off the top of my head, but we're using pyrene NHS. This NHS group is quite reactive. It'll basically do a sort of click type chemistry and bind to things like carboxylic acids. But the pyrene group sticks well to carbon or graphite, and the NHS then can be used to bond to other molecules, let's say, in this case, an antibody. We did block the surface with chitosan in this particular case, and we were able to follow that functionalization using cyclic voltammetry. So even if you're doing a SAM layer, the SAM piece of chemistry, you should be able to follow the functionalization by cyclic voltammetry, that the cyclic voltammetry should become quite distorted as you build up these layers. And if it's not distorting, then you're not really functionalizing the surface. Now, we use cyclic voltammetry a lot. I mean, I know people are using all sorts of analytical techniques. And in part, they're using lots of analytical techniques because actually they're being forced to by the reviewers. Because there was actually... It's very important to start distinguishing yourself in your kind of biosensor research, because somebody gave me a statistic the other day that 25,000 papers per year are being published. So you do need to distinguish yourself. Or in one way, the reviewers are actually just getting you to put more and more analytical work in. Of course, a slightly different tactic is actually to go to a higher technology readiness level, which is what I'm trying to promote. So at ZP, we won't necessarily do a lot of analytical work other than doing cyclic voltammetry to show that we are functionalizing the surface. Because in part we're actually, commercially focused, so we can't spend a lot of time characterizing. We want to make it and see if it works, and if it doesn't work, we're going to go back and iterate again. When it comes to testing, this is the bit that might be interesting to you, that we're testing by actually having a soluble redox probe in the solution. In this case, it was ferri cyanide. And the ferri cyanide can get to the surface, but when you get binding, you start to change the surface. Now, this was unusual because we're actually binding, and we were, in this particular paper, we're using differential pulse voltammetry, though these days I would actually use square wave voltammetry. Just my preference has changed. I mean, they're both perfectly good, but in particular as well, we've actually implemented square wave voltammetry on the sensor or potentiostat. Anyway, with the binding, the signal actually goes up. It's the magnitude that's going up here. It just happens to be a negative current because we're actually doing the reduction of ferricyanide to ferrocyanide. But the signal's going up, which is quite unusual. And I wrote the reviewer questioned that, which is a good question because it's essentially acting like a blocking assay. But I was arguing that you had ferricyanide, which is negatively charged, which is possibly being repulsed by the antibody, which may have had some negatively charged residues. But when you block the surface, essentially neutralized the negative charge on the surface, and the ferricyanide was able to access the surface easier. I don't absolutely know, though that's true. But in fact, I could have tested if that theory was true by trying an oppositely charged redox reporter like ruthenium hexamine, or even investigated it further by using a more neutral molecule, maybe like methylene blue. I was thinking of ferrocene, just as I was thinking, but when I realized with ferrocene, it's a bit unusual because he's actually got Fe2 plus sandwiched between these two. I want to say CP minuses. It's cyclopentadiene moieties, but I didn't prove it in the paper. I just made the statement, but I could have proved it by changing the charge on my redox reporters. The reason I bring this up is because it's quite a simple assay in the end. That once you've got the binding, I didn't have to then bring in a secondary sort of probe and let that bind, or reporting probe rather. I just used soluble ferricyanide. So it's worth having a look at that and seeing if it looks interesting. And then we were able to get a signal versus concentration out of it. Yeah, the problem that we're trying to solve a lot of the time, sometimes with my simpler assays, sometimes with my comment about using carbon rather than gold, and then obviously with my biased comment that people should use the SensiOil platform, is there are many papers, and Richard Compton is somebody I know who's the author of this particular paper. We even commercialized this particular paper, but a lot of papers end up finishing really at TRL3, and I think one of the routes to being able to get more sort of higher publications in higher impact journals is actually to take the technology to a higher technology readiness level, which I think the SenseIt platform actually achieves. You'll see us talk a lot about this valley of death at ZP. So I've just been talking about making RNA detection just of in the last sort of 10 minutes, but actually in the end it all comes down to, if we're going to do this, we need to kind of cross this valley of death, and this valley of death is between really technology readiness level number 3, where most scientific papers finish, and something at a much higher technology readiness level which can actually leave the lab and make an impact. And governments have been trying to solve this problem of the valley of death for years, and I think at ZP we've just been aware of it, and I think subconsciously thinking about it for years, and now we've actually got something that in part helps that. Screen printed electrodes from ZP, I'll put this link underneath the video. So a lot of what I've talked about is employing ZP screen printed electrodes, and also the SenseIt potentiostat. This is now question number 3. So this was I'm not on ResearchGate an awful lot, but when I do go up there, I sometimes find that people have asked me a question. Best place to find me is actually on Zimmer and PEDOT website. We've got a contact us page. But to be fair, this gentleman had really laid out his data very nicely and made it easy for somebody like me to, quickly read it and give him some advice. The problem that he's kind of having is, this is a bare, probably glassy carbon electrodes. He gets nice, and he's probably using a solution of ferro or ferri cyanide, and he gets nice cyclic voltammetry. Great. He's probably doing this on a disc electrode. Then he's got a graphene electrode, which I find actually quite interesting, because people try to, claim, especially I think more like commercial companies, that graphene is this wonder material and that everything's going to be great with graphene. On the face of it, actually, the graphite electrode here was actually better than the graphene electrode. The peaks are closer. I don't mind about the closeness of peaks or the distortion of cyclic voltammetry. I don't mind, because I'm a pragmatist that's trying to make real products. But if you're interested in the sort of the purity of the science, then you would actually describe the glassy carbon electrode as being better than the graphene electrode here. Why would I make that comment? Because the peak-to-peak separation is narrower on the glassy carbon electrode. I think I estimated it was about, that's about 100 millivolts, that's about 150 millivolts about, so that's about 200 millivolts. That's almost a perfect separation. It's more distorted on the graphene, which I don't mind, but it is more distorted, they're more separated. And he's using a very similar setup, I suspect, so you're always arguing that you're getting more current on the glassy carbon electrode. Now then, I think he may have done an electropolymerization of luminol to polyluminol, but then that literally the signal just gets completely died, or killed rather. I might actually have I just realized I never noticed it before. There might be a slight wave here and a slight wave here. Very slight, I know, but if I was to zoom in, I may actually find there's a slight wave here. Now the question is his sort of rationale, let's say, is, why am I able to get my ferrocyanide signal, but then when I coat it with this conducting polymer and test with my redox probe, I don't get my nice cyclic voltammetry anymore. And it was also good enough to actually send me the EIS, the electrochemical impedance spectroscopy. I have a suspicion you're using a CH instruments potentiostat, actually, when I look at this, but maybe I'm going too far there. But what's interesting is the one that I said had the classic nice cyclic voltammetry also had the the smallest charge transfer resistance. So if I just delve down into the data down here, it's very hard for me to see, but I would almost estimate that your charge transfer resistance is about 100 ohms. With the one that's your graphene, which I'm arguing here is a bit more distorted, I would super argue that, in fact, you've now come up quite considerably in your charge transfer resistance. That charge transfer resistance, I'm only able to estimate, is about 500 ohms, 0.5 kilo ohms. I can see it there. Now, when you've done your electropolymerization, so the black one, which is the bare carbon, has the smallest charge transfer resistance. The red one, which is still a good cyclic cameragram, slightly more distorted rather, has a higher charge transfer resistance. And then your massive arc here, we're looking at what's called a Nyquist plot, has a very high resistance. In fact, I can look at your, you call it arc here, I can look at the semicircle and I can actually estimate that it kind of comes out about here. I would argue that you've got something like 120,000 kiloohms, essentially. I've just doubled, you know, 60 to the top of this arc, well, to the top of the arc is about 60 kiloohms. So to complete the arc would be approximately 120 kiloohms. So you've got a massive resistance there. I do understand your statement. You're thinking that you're saying, well, this has actually got a conductive polymer in it. And you've probably done electropolymerization. But conductive is a relative term. Something could be conductive, you know, and have a resistance of like 1,000 ohms. You could describe something as being conductive and it have a resistance of 0.1 ohms. So conductive is a relative term. You're not using the sensor or potentiostat, which is no problems. I find it easier to then not be able to blame the potentiostat, but I don't believe it's the potentiostat. But that said, I know that you're doing the cyclic voltammetry of ferri cyanide on your electrode, and it's all working perfectly fine. But when you then do the luminol down, it may be conductive, but actually in your case it's acting more like an insulator, so you're essentially insulating it. But I think what's actually happening is, I'm not sure that let's say ferrocyanide can actually exchange electrons with ferricyanide. I think what the ferrocyanide, so your assumption is that ferrocyanide just is interacting with the conductive polymer and is exchanging electrons. But I'm not sure that you have evidence that that would actually work, just because the surface is conductive. Let me try and give you sort of another example. Sometimes, so for example, hydrogen peroxide, you can oxidize it quite readily on platinum, so it works well with that platinum surface. But hydrogen peroxide is actually quite hard to oxidize on a carbon surface, so it doesn't interact well with that surface. So you may have a conductive polymer, but the conductive polymer might not interact and do good electron exchange with your ferrocyanide. So in summary, if I could summarize this, clearly from your cyclic voltammetry, you've almost shut down the ferrocyanide activity, which would suggest that actually your conductive polymer is acting more like a insulator. You may have actually done so much cyclic voltammetry or polymerization that in fact you've built up too thick a layer. I suspect if you made the layer thinner, you might actually see the ferrocyanide activity. But at that point, I might actually argue that it's because actually the ferrocyanide is diffusing through your polymer and is then actually exchanging electrons directly with the carbon allotrope surface. I use the word carbon allotrope because you've got both graphene and graphite in here. So I think your biggest problem is that you're making this assumption that polyluminol actually can exchange electrons with ferrocyanide. At least in your case it's not, and I can sort of back that up then by looking at your charge transfer resistance for the two allotropes of carbon. I think for the graphite or the glassy carbon, maybe it's 100 ohms charge transfer resistance. For the graphene, it's 500 ohms charge transfer resistance. For this polyluminol coated electrode, it looks more like 120,000 kiloohms. So that would suggest that there's not good electron transfer between ferrocyanide and a polyluminol. Right, this is an interesting one to me actually, because I've been interested in drug metabolites and electrochemistry, let's say in the pharmaceutical industry, for really from my I did my first job in the pharmaceutical industry in 1997 as a medicinal chemist at a company called GSK. I did my PhD actually in electro-organic synthesis. So I've been very interested in the characterization of organic molecules using electrochemistry. Also, the generation of what's called metabolite for phase one. Phase one, I think it might be. I just remember there's the first generation metabolites and then second generation when they react with GSH, but I've forgotten just for a second what those metabolites are called. Anyway, I've been interested in, and in this particular case, I'm looking at a drug molecule called topotecan, looking at its electrochemical activity using cyclic voltammetry on the SenseOR platform. People are interested in this kind of work. A, you can detect drugs this way. B, you can actually indicate how readily drugs can be oxidized, and if you can determine how readily they are oxidized, you can give some prediction over their metabolic stability. And this is something that at ZP we do quite a lot of. Now, I haven't linked to it, but as in PEDOT, we definitely have screen printed electrodes that are resistant to organic solvents. So the first thing about doing electrochemistry of organic molecules is people assume that they're not soluble in aqueous solutions. So this is actually a test I'll do in a minute, but the vast majority of this solution, in fact, is a buffer. I've just helped to solubilize the organic molecule actually in that buffer. You can dilute organic molecules in something like DMSO, and then add something like take the DMSO and dilute it with buffer, so you get like a ratio of 5% DMSO to 95% buffer, and then you can actually do this kind of work. I think it's more important to do, especially if you're trying to determine the oxidation potential of a molecule, and the final application is pharmaceutical, to do it in an aqueous system than in an organic system, because otherwise you're going to get an oxidation potential in something like DMF or ethyl acetate, whatever it is. But that's really indicative of that molecule in that solvent, when really most of the time actually when you're trying to find out the oxidation potential of an organic molecule, you're thinking really about the human body. And at least the human body, the majority of solutions in the human body are high, at least quite high. They've got a certain pH, and they've also got a certain, they're mostly water, if I can put it that way, with also a certain electrolyte concentration as well. So I think it's actually, so at ZP, if you want to measure organic molecules in organic solvent on electrodes, we do have those electrodes. I'm not linking to it here. For this particular application, I think actually you'll get information, but you may get false information by doing it actually in organic solvents, and I think you actually need to make an effort to get it into the aqueous phase, and it's not that hard, to be honest with you. We do it quite a few times. So here we're actually, I'll do this as a demonstration in a minute, but here I can see the oxidation potential for this molecule. The peak oxidation potential is here. You can sometimes use the halfway potential as well, also as a sort of indicator of what the oxidation potential for this particular molecule is. A lot of organic molecules, by the way, I showed topotecan earlier on, they have quite complex cyclic voltammetrograms. It's easier for me to support people if they're actually on our platform, especially if they want me to start interpreting cyclic voltammograms. But that said, I might just do a quick demonstration at the moment. I realize I'm going to do a quick demonstration. I've been talking for so long, everything's actually shut down on me, but we'll see what we can do. So I'm going to do a quick demonstration where I'm going to show you how to measure the redox potential of an organic molecule. In fact, that electrode is, I think, clean. The nice thing about using screen printed electrodes for this is, the one thing about organic molecules, especially pharmaceutical type molecules, is they can be very valuable. I mean, we used to do, I used to do a lot of synthesis in the med chem industry, and the effort that would go in to make a few milligrams. In fact, I've got some customers who are in billion-dollar pharmaceutical companies doing this kind of work. And what they really like about what I'm just showing here is actually they don't have to use that much material. That was just 50 microliters that I put down on that particular sensor. I'm just going to run the assay now. So everything runs off my phone, by the way. So just do that. Done. Go to assay. Start measurement. All right, let that proceed. Right, so let me just do a quick sort of summary. So this is slightly unusual for ZP. With a lot of times we talk about biosensor development, biosensor manufacturing, wearables. Here we're talking about characterizing organic molecules that are interesting to the pharmaceutical industry. A lot of organic molecules, for example, they would undergo metabolism. An indicator of your sort of propensity to oxidation by the metabolism is actually your oxidation potential. Hey, one of the best ways of measuring oxidation potential is electrochemistry. But if you're going to do it, I think you shouldn't be doing it in organic solvent. I would actually be doing it in a aqueous system. I can understand that if you're going to do an electrolysis, and I'm pretty good at electroorganic synthesis, which is really electrolysis. You may actually do that in organic solvent, and then some of what I'm saying is not correct. But if you're trying to do oxidation potential, I would suggest that an aqueous system is a better one. Now I've got a result on my phone, and many of you who are used to Zimmer and Peacock will know this, but my phone actually just uploaded the cyclic voltammetry actually to the cloud. So I'm just logging into my cloud system at the moment. Let me just do that. So I will just refresh. Here we go. Right. So I'm just going to share my screen with you. Let me do that. External screen. Sorry, here we go. All right, cool. So I'm just showing you my screen because in fact I just did a quick cyclic voltammogram, but then I can sort of come along here and I can see my peak potential here, which is about 713 millivolts. I've actually got the second scan as well. On the second scan, it actually shifted. I know it shifted because the electrode got modified during the experiment. This is a slightly complicated voltammogram where actually I've got not only the original oxidation, but then I've got a chemical reaction and then further electrochemical reactivity. But what's quite nice about this is that this system has what's called a data pipeline in it. So I can actually tell it to go and find me peaks and report peaks back to me. So for example, if you can actually say, well, look in this window, center around 250 millivolts in my case, and open a window up of 300 millivolts. And I realize you can't see it because I've got something in the way, so I'll just shut that down. So it has a very good way of doing automatic and analysis on the peaks and able to report back peaks. So in this particular case, I actually did peak height, but you could also have done peak position as well. So if I was just to summarize what we just did, we took an organic molecule. It was in an aqueous solution. I used it on one of our definitely lower cost electrodes, and I only needed to use 50 microliters, which really preserves the amount of material that you actually need. The data automatically went to the cloud, and in the cloud actually I can do all sorts of analysis on it. And this also has an API built into it. An API means then you could pull this data across into other databases, so it's not just you don't have to manually start pulling data across or manually cutting and pasting data as well. So I hope that was interesting for people who, and I know it's a very subset of people who are interested in characterizing organic molecules, especially when they think that they're not necessarily soluble in aqueous solutions. They probably are. You just need to pre-dilute them first of all in DMSO. And it's probably worth saying that in that particular case, when I just did that, let me just go back to that. That organic molecule was not in super high concentration. It was actually about 536 micromolar, which is about half a millimolar concentration as well. Right, I will go back to my presentation. All right, on we go. This is just FYI, somebody's been very interested in connectors, and to be fair, we do actually have a nice distributor of some of ZP technologies. When I say distributor, they primarily they do a potentiostat themselves, so they only distribute screen printed electrodes, but they've been doing some work. At ZP, we do have the thing I sometimes struggle with is people don't do good connectors to screen printed electrodes. A screen printed electrode really is only as good as it's as good as the potentiostat, and it's only as good as the connector. So we do have some connectors at ZP, depending if you've got if you've got a third-party potentiostat with a 4mm banana plug, I have connectors for it. And if you have a potentiostat with a 2mm banana plug, I do have connectors for it, and I'll link those here. But a particular company called IO Rodeo in the US has actually just designed a... they got these designs online. They made some 3D printed connectors for ZP electrodes. And they've actually been doing some cyclic voltammetry, to be fair to them, on our electrodes. So it's nice because this work is not ZP work. This is generated independently by a third party. These guys are quite neutral. You know, they don't really... What I mean by that is they sort of... You know, they distribute quite a few products out there and make their own products. And so when they sort of do the work, they're doing it in a very objective manner. It's not like getting data from the company that otherwise have made the technology. So, but they have designed these connectors, and they've actually been recording on our hyper value carbon electrode and also on our 501 carbon electrodes. And it was interesting actually because they also compared against standard disc electrodes. And this is the reason I sort of use this image slightly, is this is really a good summary of the problem that but at Zimmer and Peacock, for example, let's say I was doing the characterization of a drug molecule. This particular electrochemical cell really demands a lot of solution. You can't do you can't do a lot of drug screening at this kind of volume. You'll end up consuming thousands of pounds of effort and time and money doing that. Well, of course, you can do very quick experiments on a screen printed electrode. If I'm thinking about my biosensor colleagues, when you use cells like this, it really leaves you thinking that, you know, or leaves your technology in a technology readiness level number 3, where in fact when you're using, you know, platforms like screen printed electrode and the Sense It All, then you're really leaving your technology at a much higher readiness level, like technology readiness level number 7 or something like that. But that said, they did some cyclic voltammetry using ferri cyanide on our screen printed electrodes and compared it against a glass cell. I thought it was kind of nice that they have done that work, so thank you to them for that. Right, we're getting close to the end now. This is the penultimate question, I think. Somebody wants to measure the pH in concrete. I must admit, I've been thinking about this. It was pre-2019 that I first sort of kind of started thinking about measuring the pH in concrete. So you and I are the two people who are probably thinking about this. And we do have some flexible pH electrodes, and I'm sorry that I have not linked to them in this webinar. But we do have some flexible pH electrodes. We do have the electronics to drive them. And in fact, I might have linked to that, but I might have not linked to the... I might not have linked to the pH electrodes themselves. Now, what I'm expecting is that you'll get a millivolt signal versus time. But of course, what's that connection then between millivolts and pH? And I'll have to discuss that now. You will, in the early days before the concrete is cured, you will have to independently measure the pH. I understand that as the pH actually cures, you cannot no longer then get probes into that concrete. But the reason I bring this up a little bit is because the signal that will come out of a system like this will really be millivolts, but you're actually interested in pH. But if you make 2 recordings early on, you can essentially do a 2-point calibration so that we can then convert the millivolt signal into a pH signal. So that signal is proportional to an offset. So signals for a pH probe, including the flexible ZP pH probes, are proportional to an offset plus about 59 millivolts times the pH. Now, this is really a sort of simplification of the Nernst equation. So signal is proportional to the offset and 59 millivolts, which is essentially the sensitivity times the pH. But I just want to highlight that 59 millivolts is actually temperature sensitive. So you will also have to put a temperature sensor in there as well, because I think your signal will have both a pH contribution, which is what you want, and also a temperature contribution, which you will have to essentially characterize. I haven't done this piece of science, so I'm giving you an overview of how I would do it, but I'm not necessarily able to bring you down to the and voila, it will work moment. But I think if you can measure at the beginning two pH measurements independently, that would be your 2-point calibration. And at the same time, if you can grab temperature, that would be super useful as well, because then it will help you deconvolute temperature effects from pH effects. This setup is a good start, but you've got to do. Nobody, I think, has really done this work. Is my quick scenario on this. Just lastly, and I'm sorry it's taken so long. I wonder how long I've actually been speaking for, but it does feel like does feel like quite a long time. With that said, somebody's interested in our gold microelectrode arrays, which will be mounted on a PCB. If somebody ordered them, when will they arrive in Turkey? Turkey's one of those countries that I can't predict. We can ship in 3 to 4 weeks. What I can't predict is Turkey customs. Turkey is a very export orientated country. They like to export, they don't like to import. So I cannot predict the Turkish customs. I really can't. And anyone we're shipping to in Turkey has to be very involved, talking to, because at some point they'll come to customs, and customs will be full of questions. If you want to activate that silver, then I would use the KCl saturation method. You talk here about recycling and cleaning. These electrodes are made by sputtering techniques. You've got to be very careful. We haven't tried to clean off layers and reuse these microneedles. You will be one of the first people to try that. So I cannot recommend, because we have not done it. I don't have a protocol for that. You've watched an old video of ours, and in that old video you saw that we had an old electronics. We have actually got new electronics to drive this kind of work, and you will find a link here. But I hope you go ahead. Like I say, I mean, these are fairly robust, but we just haven't tried cleaning them and taking off old layers, especially if I'm putting down a glucose oxidase layer. I find that very hard to imagine. You know, I'm worried about you scratching it and obviously abrading the surface, so I can't recommend it because I haven't done it. It's really that simple. Questions? So with PEDOT, you can easily get PEDOT onto a screen printed electrode, getting it in the microfluidic channel. Maybe if you sent me a pencil sketch, it might help me a little bit more. I hope I've given you a lot of background information on the detection of oligo, detection using oligo capture probes. I think with your polyluminol, your whole experiment, or question number 3, is based on the assumption that polyluminol will interact with ferrocyanide. I'm not sure that I was able to find a reference for that. In your experiment, it's acting just like an insulator, even though I know it's advertised as a conductive polymer. It's still conductive, it just seems to have a charge transfer resistance of 160,000 kiloohms. If you're going to measure the oxidation potential of organic molecules, maybe what they call APIs, program interface, active pharmaceutical ingredients or precursors to active pharmaceutical ingredients, and you're interested in topics like metabolites, you may have to get this into actually an aqueous solvent. You can do a dissolving in DMSO would be a good idea for that. Connectors, so you can find on IO Rodeo some actually 3D printed connector designs, so you might want to take a look at that. And then also they did a nice comparison between our screen printed electrodes, which are very scalable and something that you can take into the market. And don't forget, you know, if you're trying to get, if you haven't got much solution, a screen printed electrode is a great solution. So that we did a comparison with glassy carbon electrodes there, pH electrodes for concrete. We do have flexible pH sensors. We do have electronics. Nobody's ever done those experiments, so good luck. And with the gold PCB, yeah, we do have gold microneedle PCBs. We have not tried recycling them, and I certainly can't comment on, like I say, the economy in Turkey is definitely engineered for export rather than import. So when you try to bring technology in, I can't predict when things will arrive, because I can't predict the nuances of Turkish customs. Okay, when I say customs, I don't mean, what I mean is the actual people on the border who do controls. All right, if you've got any questions on ZP, please don't hesitate to reach out to us. Okay, thanks very much.